In the world of tumor immunotherapy, we often picture cancer cells as crafty fugitives and the immune system as disciplined police. Yet a deeper problem lurks when cancer cells downregulate MHC-I and wear PD-L1 like a protective shield. They hide from immune detection. This challenge, elusive antigen presentation, limits the immune system's precision in targeting tumors. A question emerges: could we flip the script and persuade cancer cells themselves to reveal their antigens to the immune system from within? Nature's study from January 7th introduces intratumoural vaccination chimera, iVAC, a molecule that co-opts cancer cells into becoming APCs. iVAC is a trifunctional bridge. Glue body locks onto PD-L1, an internalization baton, and a payload of antigenic peptides. Glue body, a nanobody-based glue created via Sufex chemistry, forms an irreversible bond at the PD-L1 site. Optimized fluorinated FSY variants enhance the covalent interaction, with 3,5-D-diff FSY showing peak reactivity. Once bound, this complex is dragged into the cancer cell interior, effectively blunting the tumor's defensive PD-L1 armor. Inside, a Trojan horse mechanism unfolds. CPP pulls the complex through the endosomal pathway toward the lysosome. PD-L1 degradation in the lysosome is rapid and profound, dramatically reducing the immune suppressive shield. But to present antigens, the payload must escape into the cytosol and reach the endoplasmic reticulum for MHC-I loading. This escape relies on perforin 2, whose expression increases after iVAC treatment, enabling antigen trafficking to MHC-I. Fluorescence resonance energy transfer tracks the journey. Antigen is liberated in the lysosome, then escorted to the ER, bound to MHC-I, and surfaced. Immunopeptidomics confirms many exogenous peptides on the tumor cell MHC-I, a sign of successful cross-presentation. The tumor now presents foreign peptides on its surface, shedding the PD-L1 shield and signaling to T cells. In mouse models, iVAC turned cold tumors into responsive ones, achieving complete remission in MC38 tumors. T cells infiltrate, activate, and secrete IFN-JA and TNF, signaling a robust anti-tumor response. Macrophage polarization shifts toward the anti-tumor M1 phenotype, reducing suppressive M2 and TREGs. Antigen spreading broadens the attack. Initial foreign antigen exposure leads to responses against endogenous tumor antigens. Scientists then tested iVAC CMV, pairing the chimera with CMV-derived peptides to mobilize pre-existing memory T cells. In human tumor cells, CMV epitopes appear on the surface after iVAC CMV alongside endogenous tumor antigens. A humanized mouse model with patient-derived tumor cells demonstrates that iVAC can redirect virus-specific T cells against cancer. In a broad panel of patient-derived tumor clusters, the approach shows meaningful efficacy across several cancer types. Across 28 patient-derived models, overall tumor response reached 61%, with higher PD-L1 expression predicting better outcomes. This clever mechanism makes the strongest tumors, the most immune-suppressive, most vulnerable to iVAC attack. Beyond therapy, iVAC redefines targeted degradation. It couples destruction with antigen presentation, a two-in-one strategy. It provides a general route to turn cold tumors hot without using viral vectors, sidestepping associated risks. A key insight is that iVAC does not rely on a single tumor antigen. It tags the tumor with exogenous antigens to recruit memory T cells. Clinical translation faces challenges: tumor penetration, dose control, and safety in the human immune milieu. Low PD-L1 tumors may require combination strategies to trigger target exposure before iVAC delivery. The end goal is clear: coax cancer cells to reveal their guilt, enabling precise immune action and durable memory. In closing, this chemistry-driven reprogramming of cell fate blends molecular engineering with immunology to tip the battle in the body's favor. If cancer cells are made to become their own antigen-presenting agents, the balance of war could finally tilt toward the immune system. This film highlights a future where treatments leverage existing memory T cells, turning patient history into therapeutic power. From bench to bedside, the iVAC approach questions the old dogma and opens a new path for treating solid tumors. As research progresses, the dream is clear: more patients achieving tumor control with durable immune memory,